Good afternoon, ladies and gentlemen. My name is Dr Inge Gazendam and I'm a researcher at the Agricultural Research Council. In this presentation, I will briefly introduce you to molecular breeding and give a few examples where we are applying it to 2 crops at our institute. I will start with what molecular breeding is not. For the purpose of this presentation, I interpret it to not include genetic engineering. This is a fascinating topic with various applications in plant breeding, but will require a freestanding presentation of its own to do it any justice. So what do I interpret as molecular breeding in general terms? During breeding, breeders want to combine different parts of 2 organisms' chromosomes to combine certain characteristics of each. After making the cross, you have to determine if the correct part of the wanted chromosome did actually transfer to the offspring. If it is a plant, you could grow the seed and wait to look for the desired characteristic. You wouldn't need markers if the characteristic was easily observable, example seed shape. But if it is, for example, a tree, you would sometimes have to wait for 5 to 10 years before it could bear fruit that you were trying to improve. If you need to improve yield, for example, multiple yield trials with many thousands of plants will be needed to accurately measure the improvement in yield of some offspring. This is an intensive use of resources, namely manpower, time and money, and scientists always look for ways to reduce the research requirements of breeding. So molecular breeding was developed, where we use an indirect method to look for if the gene of the characteristic that we want to transfer did transfer to the offspring. So what is this indirect method? It's molecular markers. A molecular marker is using an indirect method to identify a specific gene located on a particular chromosome. It can be a piece of DNA, a gene, then it's a genetic marker, or a phenotype expression of a gene. Since the marker and the gene it marks are close together on the same chromosome, they tend to stay together over generations. Molecular markers are therefore used to track the presence of a gene that is difficult to detect otherwise. Genetic marker is a specific type of molecular marker. It can be defined as a specific DNA sequence with a known location on a chromosome that are close to or in the gene of interest. Since they are close together on the same chromosome, they tend to stay together as each generation of plants are produced. Genetic markers is dependent on the existence of genetic variations or DNA sequences that differ between individuals or populations. These are called genetic polymorphisms. There are different techniques for detecting genetic variations, and the polymorphisms most often used for genetic markers include microsatellites and single nucleotide polymorphisms. Genetic markers can be one of 2 types. The first is gene-linked markers, which are markers developed from regions which are tightly linked to the gene of interest. The next is functional molecular markers, developed from polymorphic regions within a gene which cause the variation in the characteristic that you are breeding for. In the case of B, if you know which gene is responsible, you could determine its presence or expression directly. Example, you could look for the gene sequence or enzyme that catalyzes the required reaction. But many times you don't know the causal gene and have to find another indirect genetic feature that is associated with the characteristic you are tracking. Then you would use a marker that is linked to the gene, such as in figure A. I'm now going to give 2 examples where genetic markers are applied at ARC-NSW. The first is where we use DNA markers for onion male sterility to identify the correct maintainer lines. The second is where we use DNA markers for DNA fingerprinting of potato and other crops. For the first example, the objective is to use markers to identify the correct onion male sterile and maintainer lines to supply to seed companies for hybrid development and commercialization. To determine the male sterility of onion parent lines previously, they had to be planted and grown before it could be checked for pollen sterility. This had to be repeated over multiple seasons for accuracy, and there were many difficulties experienced during scoring. First, for some theory on what cytoplasmic male sterility is. It is a naturally occurring genetic condition in onion and many other plants, which plant breeders have harnessed to prevent self-pollination in order to produce hybrids. In the nucleus, there is a nuclear restorer of fertility allele, capital MS, but we are only interested in the homozygous recessive small ms plants. The cytoplasm can be one of 2 types, normal or sterile. This is not inherited from the male plant since the pollen grain doesn't transfer its cytoplasmic genetic material when fertilizing the ovules. These two work in combination to result in plants that are either normal, with functional pollen, as seen in the onion flower on the left, with yellow stamens sticking out above the white flowers, or male sterile plants on the right. The male sterile flower does not produce pollen, but this is sometimes difficult to score if you look at the flowers of the wrong age. When they are too young, you cannot see the difference. If you wait too long and the pollen was produced, you are too late to remove the wrong plants, and it may already have been transferred by the pollinators, which are bees. The method for screening of onion male sterility using markers is the following. You first extract DNA from the leaf or bulb material, depending on what stage of the planting cycle the plant material is that you want to test. For the PCR reaction, the onion DNA, buffer, the NTP, the polymerase enzyme, and the marker primers are all added together in a tube. The temperature of this tube is then cycled in a pre-programmed thermocycler to amplify the DNA. Afterwards, the fragments are separated on an agarose gel with electrophoresis, visualized with UV light, and then a photo is taken of the gel. But this is a special kind of PCR called a multiplex PCR assay, where we combine the different sets of primers in the same tube so that you can detect the nuclear and cytoplasmic genotypes at the same time. For the nuclear gene, we use the ACSKP1 marker, which gives the capital MS or small ms genotype. For the cytoplasm type, we use the ACCD marker, which tells us if the plant has sterile or normal cytoplasm. The result is, on a single gel, you can detect the genotype at both the nuclear and cytoplasmic male sterility genes. The advantages are that it is fast. You could determine the onion genotypes within 1 day of isolating the DNA. And you use less reagents, because only one PCR reaction and one agarose gel is required for both nuclear and cytoplasmic genotyping. Using DNA markers for male sterility screening is therefore of vital importance, because it enables the onion breeding program to recover the correct genotype of South African onion hybrid parental lines, and it facilitates the production of the correct parent seed for commercialization of ARC onion lines. The second example where genetic markers are applied at ARC WIMP is for the DNA fingerprinting of potato. Potatoes is an important commodity in South Africa, and producers often import superior germplasm. All role players expect potato production to be of the highest quality. The objective of DNA fingerprinting, therefore, is to identify genetic differences between cultivars, to confirm trueness to type of potato cultivars, and to identify cases of accidental mixing. The current method for DNA fingerprinting is the following. DNA is isolated from any part of the plant that is sent in by the client. It could be leaves, tubes, or small in vitro plants. The DNA fingerprinting relies on the technique of detecting simple sequence repeats or microsatellites with genetic markers. SSRs are tandem repeats of short motors, for example 1 to 6 nucleotides long. There is a variable number of these repeats in different cultivars due to incorrect replication or slippage of the DNA polymerase during replication. SSRs are also dispersed throughout the genome. The markers used for the SSR primers are primers that bind to the unique flanking regions on either side of the SSR region. When you use them in PCR, they amplify pieces at different lengths, called different alleles. SSR for fingerprinting is a PCR-based technique where the template DNA, the buffer, the NTPs or the building blocks, the polymerase enzyme and the flanking primers are all added together in a tube. The DNA fragments of different lengths are then amplified by cycling the temperature in a pre-programmed thermocycler. The fragments are separated on a polyacrylamide gel with electrophoresis and stained with silver staining to reveal the banding pattern. Before the gene bank multiply cultivars for clients, they have to check if the clones for a particular cultivar are all correct. Often thousands of small plants are multiplied starting from just 5 initial plants. For this trueness to type determination, the result of the SSR indicates that for cultivar 1, all the clones look similar, therefore there is no problem. But for cultivar 2, one of the clones differs. Therefore the wrong clone should be discarded before multiplication proceeds. Using DNA markers for crop fingerprinting is therefore of vital importance, because accidental mixes can easily happen even with proper precautions. When mixes occur, it can be identified at a very early stage using DNA fingerprinting to avoid big costs later. For example, when a whole field is planted with potatoes of the wrong type, and it cannot be used by the processing industry. Thank you for your attention.